Welcome to the last part of Introduction to Nutrition. In this video, we're going to take a look at food tests. So this video is basically about a practical part of biology, and of course the question also comes in paper 1 in multiple choices. What you're going to do here, some of you are lucky, they may be part of your practicals. So the job is just to observe the steps and what happens. So that's the whole point of this video. So we're going to start with the iodine test for starch. How do you test if a given food sample has starch? So you use what is called iodine solution, and the test is called the iodine test. So this test can be carried on different types of food samples. The first is the food sample that is in solid or powder form. So if you have a food sample which is in solid form, like a piece of sausage, or one that is in powder form, like a piece of spices maybe. So how do you test if there is starch? So you place a small amount of sample on a white towel. You add a few drops or 2 drops of iodine solution to the sample, then observe and record what happens. So you get your towel here, you put the food sample to be tested, you get your iodine solution. So you add a few drops or 2 drops of iodine solution, then observe what happens. If there's starch, this is what's going to happen. The food sample the mixture of iodine solution and the food sample will turn blue-black. If this happens, then know that there is starch. If nothing happens, no changes, just know that there is no iodine. There is no starch. Iodine is brownish in color, so when you add iodine to your food sample, there might be some color changes where your food sample will appear a bit brownish, or depending on the color of your food sample, there could be some color changes. But so long as it's not blue-black, then your solution doesn't have starch. Then the second type of food samples are samples that are in solution or suspension form. How do you deal with these ones? So the procedure is nearly the same. The first is you place 2 cm³ of sample solution into a clean and dry test tube. Then you add a few drops or 2 drops of iodine solution to the test tube and shake. Then observe and record what happens. So you have a test tube here. You need a measuring cylinder where you're going to put your 2 cm³ of sample solution. Then once you have that, you can pour that solution in here. So this is going to be 2 cm³. Then you can add a few drops or 2 drops of iodine solution. Then observe and record what happens. If there is starch in this food sample, it's going to turn blue-black. But if there's no starch, nothing is going to happen. Or it can have some funny color changes. That's because of the brown iodine you've added, but it won't turn blue-black. The second test is the Benedict's test for reducing sugars. So this test is used for both reducing sugars and non-reducing sugars. Some of the misconceptions that some students make is if they test for reducing sugars and they discover that there's no reducing sugars, they automatically think that there is non-reducing sugars. Non-reducing sugars is a different nutrient as reducing sugars. So if you test for reducing sugars and you discover that your food sample does not have a reducing sugar, it may have other nutrients such as starch, proteins, but it doesn't necessarily imply that there is non-reducing sugars. So how do we test for reducing sugars and how do we test for non-reducing sugars? So the procedure is nearly the same, but anyway, let's start with the test for reducing sugars. So this test requires the sample to be in solution form and may be performed on suspensions. So what does this mean? It means if we have a food sample that is in solid, you need to grind it, then you mix it with water so that it dissolves in water, and then that's when you can do this test. So step 1, place 2 cm³ of sample solution into a clean and dry test tube. Add 2 cm³ or just an equal volume of Benedict solution to the sample solution and shake. For example, if you haven't placed 2 cm³ of a sample solution, you have placed 3 cm³ of sample solution, maybe by mistake, just add the same 3 cm³ of Benedict solution to the sample solution and shake. After that, gently heat the mixture using a water bath, then observe and record what happens. So if this is your test tube, you first have to add a 2 centimeter of a sample solution. Then you have to add an equal volume of Benedict solution. Of course, the way I'm drawing it, I just want you to see the different solutions, or the different I want you to see the food sample mixture and the Benedict's solution. But of course, when you add, they are going to mix, so they won't appear like this. Then after that, when you gently heat the mixture using a water bath, if this happens, it means there is reducing sugar. So if the entire solution turns brick red, then there is a reducing sugars. So in summary, if your solution remains blue, then reducing sugars are not present. If your solution turns green, yellow, orange, brick red, then your reducing sugars are present. So these are the different types of colors that your solution can turn. You have to write the final color, not like my solution turned green, yellow, orange, brick red. No, you have to just write your final color. So the different colors proportionate or correspond to the amount of reducing sugar in the solution. So if your food sample has little reducing sugar, you might just observe green. If it has a bit more reducing sugars, you observe yellow. A bit more than yellow, the observation is going to be orange. If it has plenty of reducing sugars, then you're going to observe brick red. So in your answer, you just have to indicate one color. So that's how to go about it. So continue to the Benedict test for non-reducing sugars. So when you are done with testing for reducing sugars and you discover that there is no reducing sugar, you might need to test if there is non-reducing sugars. So what you do again, you start the procedure, but before you can start doing the test for reducing sugar as prescribed in the previous methodology, what you have to do is you have to place first another 2 centimeters of a sample solution into a clean and dry test tube. So this is a test tube. You place your food sample, which part of it you tested and you discovered that there is no reducing sugar. Then you have to add 1 centimeter cubed of dilute hydrochloric acid. Then you have to heat the mixture for 3 minutes and then cool. So you add 1 centimeter cubed of dilute hydrochloric acid, you heat in a water bath for 3 minutes and cool. Then 3, you add sodium hydrogen carbonate solution or sodium hydroxide solution to the mixture, a little at a time until fizzing stops. So when you add sodium hydrogen carbonate solution, the entire solution is going to be to start fizzing. So you add another, it will keep on doing that. You add another, keep on adding until it stops fizzing. Once that is done, add an equal volume of Benedict solution to the mixture. So you add an equal volume of Benedict solution to the mixture. When you do that, you gently heat the mixture again using water bath, then observe and record what happens. So when you observe and record what happens, you might have a situation where this happens. The entire solution turns brick red. So when this happens, when the entire solution turns brick red, then you have non-reducing sugars in your food sample. But if nothing happens, then if you don't have brick red, then you don't have non-reducing sugars in your food sample. So if the solution remains blue, non-reducing sugars absent. Solution turns again green, yellow, orange, or brick red, non-reducing sugars present. Again, if the solution turns green, it means there is just a bit of non-reducing sugars. If it's yellow, it means there is a bit more non-reducing sugars. Orange, again, we have more of non-reducing sugars as compared when it was yellow. Then brick red, it means we have plenty of non-reducing sugars. We go to the Buell test for proteins. This test also works best for solutions and suspensions. So again, the procedure is you place 2 centimeters of a sample solution into a clean and dry test tube. So I have a test tube here, put your 2 centimeter cube of a sample solution. Then here you can do either of these depending on what you have been given. If you haven't been given a burette solution, this is what to do. You add 5 drops of sodium hydroxide solution to the sample solution, followed by a few or 2 drops of copper 2 sulfate solution, drop by drop, shaking and observing after each drop. So you add 1 drop, you shake and you observe. Again, you add another drop, you shake and observe of any changes. If you haven't been given sodium hydroxide solution and copper 2 sulfate solution, you add an equal volume of burette solution. So burette solution is already made up solution, unlike these two which you need to add one by one. So when you add an equal volume of burette solution, which is this, and if you end up with a purple color or a violet color, then you have proteins in your mixture. So if your solution remains blue, you don't have proteins. If your solution turns purple or violet, then you have proteins in your mixture. So for those who are color blind like me, purple is this, violet is this. But don't worry, like asking yourself if it's violet or purple. Just record what you can observe. If your eyes can tell that it's purple, record that you have seen purple. If your eyes can tell that it's violet, then record that you have seen violet. We go for testing for lipids. Lipids are of course fats and oils. So there are 2 tests that are done on lipids, and this is going to be our last test. So we'll start with the emulsion test. So in this test, you shake a small sample or a drop of sample solution with 2 centimeter cubed of ethanol in a test tube. Then add a few drops of distilled water to the test tube, then observe and record what happens. So when you do this, if you have lipids in your mixture, then these are the possible outcomes. If your solution remains clear, then you don't have fats or oils in your sample. If an emulsion is formed or a solution turns cloudy, then you have fats or oils present. So an emulsion, the way you can tell an emulsion or cloudy is it's not as clear as the solution you had. So I'm talking about something like this. So this is an emulsion or a cloud. So here down here is clear, but here there's an emulsion. So if your end results are like here, where it's where you can see the behind of the test tube, then you don't have fats or oils. But if you have this situation where you can't clearly see behind, then you have fats or oils. The second test for lipids is called the grease spot test. So in this one, you place a drop of sample or sample solution on a filter paper or brown paper. So this is a brown paper. You put your food sample here, which you suspect to have lipids. You place a drop of distilled water next to the drop of the sample. So get a drop of distilled water. You make sure that it is there and then mix. Then hold the paper against light until the drop of water disappears. Then observe and record what happens to the sample spot. So when the two mix, then when you do the step 3, then if your solution forms a permanent translucent spot, then you have lipids in your food sample. So if your sample spot disappears, fats or oils are not present. If your solution forms a permanent translucent, oily or grease spot, then your fats or oils present. In the in real world, this is what you can see. So these are examples of permanent translucent spots. So translucent means you can't see through. So this is how to do the grease spot test for lipids. This marks the end of our lesson. Hope you're going to make use of this the information that has been shared in this video when you're doing practicals. Otherwise, happy solving.